Hi, everyone. Welcome to another postdoc webinar. If you've been here with us before, welcome back. If this is your first time, I'm just going to give you some info real quick. My name is Rina Dukor. I am the president of FAU's Postdoc Association. And this webinar series is co-organized by the Office of Postdoctoral Affairs at FAU and also by the FAU Postdoc Association. So you probably noticed that we're on a webinar mode, so your microphones and cameras will not be available. However, we do want to hear from you. We want you to interact and bring your questions. So feel free to type in your questions using the Q&A button at the bottom of your screen. If you type in your questions during the talk and our speaker is still talking about that topic, I can bring the questions in right away. But if not, we're going to have a Q&A session at the end of the talk anyway, so feel free to type in your questions at any time during the talk, and our speaker will address them at the end. Okay, so without further ado, it is a pleasure to introduce our speaker for today, Dr. Rina Dukor. So Dr. Dukor earned her Ph.D. at the University of Illinois. And 20 years ago, she left her postdoc position to launch BioTools, which is an instrumentation company specialized in characterization of chirals and biological molecules, and is located here in Florida. So celebrating the 20th anniversary of her company this year. Dr. Dukor has also put a lot of effort over the years in supporting women in science and mentoring young scientists. So she's also very passionate about mentorship. So without further ado, welcome, Dr. Dukor. Thank you for joining us today.
Thank you, Lorena, very much for your very kind introduction. And thank you, Jenny, for invitation. And thank you, everybody, for joining us. Should I go ahead, Lorena, and proceed? Okay, perfect. Just let me then share my screen. It is definitely a pleasure to speak to a postdoctoral association and postdoctoral and graduate students. As Lorena mentioned, I am very passionate about young scientists and specifically, of course, women in science. And if you go to microdomain, immigrant women in science or any immigrant women, because it's actually immigrants, because it's actually a little harder to come to this country and to jump into a new country and a new research, and especially from a different country. So again, thank you everybody for inviting me. I am absolutely delighted to tell you my story. It's a little hard to know how much to present and what really matters to different people. My story is not unique. Yesterday, I celebrated 40 years coming to America as a very young girl leaving the Soviet Union. And I wrote a little bit on LinkedIn about what it means for me to be in America and what the biggest difference is between where I used to live and where we live now, and how America has changed, obviously changed my life, and the dreams that, you know, when I came at 15 weren't clear. But I was able to go to university. It wasn't easy for a Jewish person to go to university back in Russia, and of course from university to PhD and so on. And that's the story I'm going to tell you. I will talk a little bit about science. I apologize in advance if it's a little bit too much science. I tried to take it down because I don't know if I'm talking to biologists or chemists, biochemists, physics, physicists, and everything in between. So hopefully this will be not too much science. Okay, so with all of that, let's just turn a little bit, just excuse me for a second. Okay, so it does work. Okay, so what are our choices? You've probably seen types of slides like this many, many times. And so what are our choices after we get PhDs or we finish our postdocs? And this is not anything new. It's just to kind of remind you of that there is lots and lots of choices. A lot of people, at least in my field, which is chemistry or biochemistry, go to industry or do as a job or industrial postdoc, or they do academic postdocs. So industry is definitely, I would say, probably number one field where most postdocs and students end up. Academia, government lab, so professorship or an academic post, either as a postdoc or a professorship after. And I, of course, want to give a little shout out to the last few women in chemistry who won the Nobel Prize, including today's winners. So Professor Frances Arnold won in 2018, and today we have a historic date of two women professors, Emmanuel Carpenter and Jennifer Doudna, winning for their CRISPR research. So all of you out there, that's what you can strive for if you want to decide to go to academia, and even in industry, you can win the Nobel Prize. But that's not the only pathways. You can, of course, go into what I call a business of science. And business of science to me is everything you do to support the science. And from company perspective, that includes marketing, law, sales, business development. There is lots of things to do to support scientific endeavors. And when we get interns from local high schools or universities, I always ask them, like, I have a lot of parents pushing their kids to go into medical school, or I see a lot of parents pushing their kids to go to medical school or scientific careers. And when they get here, I always tell them, pay attention to the business of science, because things like marketing and law and even sales, business, all of it requires a good scientific knowledge, and you can still do science and not be in a lab. So if for some reason you don't like being in a lab, you have lots and lots and lots of choices of a career that still involves your skills. There are, of course, other careers. You can do lots of different things. And then there is entrepreneurship, which combines, from my perspective, absolutely everything that's about it, 1 through 4. Because entrepreneurship, it's a different type of a career to some extent. Why is this not okay? Let me minimize this and see if this will be better. Okay, so like most of you, I graduated with my PhD and had a PhD advisor. This was my celebration. In 2 days, I will be celebrating my PhD defense in 1991, so a very long time ago, in October 9, 1991. And just like coming to America and just like getting married and having children, you never forget your PhD defense day. At least I know I didn't. We worked very hard for that day. And my professor pictured here is Professor Timothy Keireling. And on my next screen is what I call Themology 101. So when I was a student, still as I graduated from high school very fast. I was still fairly young when I entered graduate school. And what he told me at the time was the PhD program was not to learn a particular skill. I wasn't there to learn spectroscopy per se, or to learn how to build an instrument, or to learn how to analyze a protein. But it was to learn to be an independent thinker and an independent researcher. And that really stuck with me for life. This quote, I have given the same quote over and over, because it is so important to really grasp that and to say that you're now thinking outside the box, but on your own. You are the one who's coming up with questions and ideas. It's not necessarily your PhD or postdoc advisor. And to ask questions and to know where to find the answers and how to try to answer these questions. Of course, now lots of people go to Google. Back then, that wasn't the case. But how to think deep and to answer these questions, no matter what the career is. And my take on it, in addition to really understanding what that meant, was study hard, ask lots of questions, explore as much as you can, learn, publish, and the last was accept challenges. If someone says no to you, and I have heard more no's and I have heard yeses in my life, you just work harder and prove them wrong. And that's the story of my entrepreneurship and BioTools because of no's, almost because of no's. So as I graduated UIC, as I just told you, I graduated in October of 1991. But in August, I already had a job offer for an industrial postdoc. And I'm not showing this to show off, but I like to tell the story because it was one of my first real experiences of exploration and saying yes. So one day I walked into the chemistry department and there was a big notice that Amoco Research Center, which was a big facility and one of the biggest companies in the Chicago area where I lived, was looking for guinea pigs to help their new managers learn how to interview people for jobs. So it wasn't a job interview, but they were looking for people to come and help them be the guinea pigs, and they paid $75 for the day. And of course, a graduate student, and especially at that time, $75 was a lot of money. But it was an hour away, but I called the next minute after I saw the advertisement and scheduled to come to Amoco. And during my interview process, there was a woman who just became a new manager, and she was so impressed with me that's the story she tells me, that she realized what I was studying, and she passed on my resume. We had to submit resumes to a friend of her in biotechnology division. I was studying the structure of proteins at the time. So a couple of weeks later, I got an invitation to come and visit the site again. And in August, got a job offer for an industrial postdoc. So to me, that lesson is, if you see an opportunity, take it. You never know what an opportunity will lead to. By being on this webinar, you're taking the first step. That is a step because you don't know who I know. You don't know who I can introduce you to. And so building your network, learning how to interview in my case, or meeting new people, passing on your resumes, discussing. I wanted to understand at the time what Amoco did. I knew it was a big conglomerate. I heard that they had, as you could see, Amoco Technology Company, and I just simply wanted to know. I never in my wildest dreams imagined that they would be studying protein structure, but that was in fact the case. So explore and take chances is my lesson number one. The second thing I learned at Amoco, well, actually, I've learned a lot of things in Amoco, and I'll come back to the first, but leading, participating, volunteering, and networking was probably one of the most important lessons. And the best way to do this is to participate in your local and national societies. So like Lorena is doing right now, leading the Postdoc Association for FAU, or like probably many of you belong to either American Chemical Society or American Neuroscience Society, or whatever society or American Mass Spectroscopy Society, whatever society you belong. Participate in society. Don't just belong. Participating and volunteering for committees is probably the best thing you can do. There's also a lot of committees and associations right here in South Florida. We have BioFlorida, we have Bass Florida. There is lots and lots of organizations that you can join and volunteer. I'm blinking out on a couple other ones, but there is a few. And what that will give you is, it will give you experience, first of all, to work with other people. When you work in a lab, you often work by yourself. It will give you a chance to learn how to come up with ideas and implement those ideas. In my cases, I participated in the Society for Applied Spectroscopy. I learned how to compromise, which was really a big thing. I learned how to present. I learned about budgets, which I didn't have a chance to learn in graduate school, because even though we did do grants, it's not like a total profit and loss. So leading, participating, volunteering, I cannot recommend that enough for graduate students and postdoc. That is probably your best avenue to success and building your career. What I also learned is I was the second woman Ph.D. at at that, Amoco Biotech. And the first woman left shortly after, so I was the only woman PhD for quite some time until we started hiring other people. And I don't want to say that I wasn't noticed, but it definitely was a shock because in graduate school, there was a lot of women studying for PhD. So it was very strange to me to see that only so many women were in industry. Or maybe it was because I was in physical chemistry, biophysical chemistry. And how to stand out and be noticed. I have lots of stories. I won't spend the time right now, but certainly one way is to volunteer and network and lead and participate. And again, I'm going to refer to Lorena for leading the FAU Postdoc Association. That is how you stand out. Take the opportunity to publish and present. That I cannot emphasize that, not from the scientific perspective necessarily, which of course is so important for your career. You must publish and present, but it's also a must to learn your oral and written skills. All of those jobs, all of those careers that I have mentioned, including the business of science, you need all of those skills. You need to be able to talk. And one thing I like to tell people is that everybody sells. Doctors sells. PhD scientists sell. We all sell what we do. We have to sell our science to the grant agencies. We have to sell it to our peers in order to convince them that what you have just discovered is true. So everybody sells, believe it or not. And so to learn those selling abilities, or whatever you created, research, developed, is very, very important. And the only way you can do that is through oral and written skills. And your best venue as postdocs and graduate students is to do that through publishing and presenting. And the last thing, which I actually had as the first item, is always look for opportunities. Always. I started as a scientist. I was working on structural elucidation of proteins, but as I listened to customers, in this particular case, it was protein design labs, and tried to understand what their need was, I immediately saw that the science that we were doing would be tremendous as a product, because if one company is asking for help, that means the second and the third and the fourth need the same answer. So whatever tool I made, if somebody is using it, possibly somebody else might need it as well. And so I pushed the management, after I learned how to do business plans and market analysis by being in the meetings, I pushed the management to let me create a commercial product called Proda, and I will talk a little bit about that. So that job offer from Amoco in 1991 was a 2-year postdoc, which after I was done, they offered me a full-time job as a research scientist. And I spent about 6 years there, and sort of 6-9 years later, depending on how we look at it, I started a company called BioTools. And as Lorena mentioned, we are celebrating 20 years of the company, which is almost hard to believe, but it's absolutely fascinating. And before I run out of time, I want to say thank you to everyone who has played any part in the company, and that includes foremost employees, customers, collaborators, vendors, lots of interns, especially here in Florida, and lots of students. Everybody makes a difference. Everybody makes a mark. It's not I am the voice, but I'm not the one who does or did all the work. So thank you to everyone who's been on my path, and especially to my co-founder, Professor Naifi, who's right there in the back. He was a professor at Syracuse University and joined forces with me to do BioTools as co-founders. So we are located right here, pretty much where some of you are, in Jupiter, right on Beeline Highway. I don't need to explain to you where we are. I like to show these pictures when I give talks, because not everybody, you guys probably seen lots of alligators, but a lot of people don't. We are also an international company, even though we are right here in Jupiter. We have an office in Europe. We have a network of distributors and collaborators all over the world, including Asia. We have centers for what we call global centers for chirality, again in Europe, China, Brazil, India. So this starting the company and being an entrepreneur gave me a tremendous amount ability to travel. So why did I need to start a business? I had a great job at Amoco, great salary, the job that I loved. Why did I need to start a business? And the reason was science, actually, and my love for what I was doing. It was not financial. It was simply the desire to let other scientists do the same science that I do. I knew at the time that there were three types of pharmaceuticals on the market. There were small organic, sort of like little aspirins. And the literature and the news at the time was filled with discussion of two other new types of pharmaceuticals. One was called chiral, and one was protein-based, or other biopharmaceuticals. And that's all you heard. The biotech industry was being born, Genentech and Amgen were being formed. And so that was all the talk in science at the time. Now, chemical and engineering news, which I'm a chemist, so I'm obviously a member of the American Chemical Society, which I just told you is absolutely critical to join the societies. And every year, Chemical and Engineering News dedicated an issue to chiral drugs. And that's been going on for years. And while I was studying proteins at Amoco and developing a product called Proto, which I just told you about, I kept reading about chiral drugs. And just to remind you, to introduce some of you from your Chemistry 101 class, in case you don't remember, chirality is a non-superimposable mirror image. So it's just like your left and right hands, where the left fits really well into a left-handed glove, and the right doesn't do the same. So the pair is called enantiomers. One of the best known examples, pharmaceutical is a drug called thalidomide. Women in the 50s and the 60s took it, and unfortunately babies were born with deficiencies because this molecule epimerizes, forms a mixture of the two forms in the body, and one form helped stop the growth of the cells. Stop the growth of the cells. The other helped with vomiting, but that's why the babies were born with deficiencies. In Florida here, we have lots of interns who've done this project. You can take an orange and a lemon grown right here. You can squeeze it out, you can separate it. And the main molecule in that fruit is limonene, and limonene exists in two mirror image forms known as R and S limonene. And you can smell it, and you can smell the notes of orange, and you can smell the notes of lemon. And actually that's really, I think one of our students has won a science fair for a similar project or a project like that. The same for carbon. It's one form smells like spearmint, the other form smells like caraway seeds. So chirality is critical. Chirality is important, and nowhere is it more important than in pharmaceutical industry. So your grandparents or your parents, unfortunately, might be taking some of this drugs. You might have heard of Lipitor or Plavix or Zoloft. And then biological side, you might have heard of Humira and Rituxan and Herceptin for breast cancer. Humira, I think, is still the #1 sold pharmaceutical in the world. So there's lots and lots of these drugs. And these days, I'm going to come back to this. Why is chirality important in pharmaceutical industry? I've already mentioned it, but the biological targets, our enzymes, are chiral. And so the molecules that interact with them have this sort of key and shape interaction. So the shape is really, really important. So chirality is critical for potency, selectivity, pharmacokinetics, toxicity, and so on, because shape matters. Some other well-known drugs are Motrin and Aleve. So ibuprofen is sold as racemate. And one form of the drug could do, just like for thalidomide, could do lots of good, but another form could have side effects, such as stomachache as an example. So when you buy something as a racemate, you could have more side effects, whereas if you buy something as a pure enantiomer, it probably has better efficacy and less side effects. And these days, chirality is all over the news, even though people don't realize it. So everybody's talking. We're living in the COVID era, in the viral era, and the flu is coming up again. There's 4 FDA-approved flu treatments, and every single one of them is chiral. The Tamiflu, Relenza, and so on. So everything, when you see dots like this, or when you see solid lines like this, that means that the molecule is chiral. So chirality is absolutely critical. And the antiviral drugs you see on TV, and especially with so many famous people getting COVID, remdesivir is a chiral molecule from Gilead. There is several chiral centers, in fact. There was another drug, miripudib. It's also chiral. The third one that's being used is not chiral. It's a very small molecule. But majority of the drugs, especially in the antiviral world, I think there's about 100 antiviral drugs on the market, and more than 50 or 60% of them are chiral. So chirality is critical. Other types of pharmaceuticals, even if so all biological molecules are obviously all chiral, because peptides, proteins, nucleic acids, carbohydrates, everything is chiral. Steroids, dexamethasone is chiral. Carbohydrates, I already mentioned that. So almost everything, we are surrounded by chirality. Either you know that or not, we are. So that was how I came about to chirality. And as I read that Chemical and Engineering News magazine every single month, once a year, and saw that chiral drugs becoming very important, I realized that my Ph.D. work, the instrument that we worked on for looking at the structure of proteins, could be absolutely phenomenal for solving one of the most critical parameters in chirality, and that's known as absolute configuration, which tells you if you've made a left-handed or right-handed drug. So that's another lesson, I guess. Always think outside. And I knew right there and then, honestly, on a plane, I sat down and wrote a business plan as I was coming from one of the meetings, because I understood that in order to determine chirality, you had to grow a crystal. And the technique the instrument that we built in our lab during my PhD study could give you that answer in solution. And so I just became very passionate and determined, not just passionate, but absolutely determined that if I could just build that instrument and I could just sell it to everybody else, then all of these drugs could get to the market much faster. And that was the reason for starting the company. I'll come back to biological molecules because I just want to stay with chirality for a second. So the technique we commercialized, there will be no test at the end, but just for those of you who are chemists in the audience, is called vibrational circular dichroism. Everything we do here is based on vibrational spectroscopy, because that's what I learned in my graduate school. That's what I did. The second technique we commercialized was Raman optical activity, and this FTIR spectroscopy of proteins is what I asked the Amoco managers to let me create as a product. Once I left Amoco, I negotiated taking the rights to that technology with me to BioTools. So right here in Jupiter, we have a couple of things. We make the instruments right here. We also now have accessories. We've expanded our product line, and we also run a contract research lab. So for those companies that are smaller, second, third, fourth tier, or startups, or academics who cannot afford their own instrumentation, we help them solve absolute configuration. And in fact, I was going to put this, and I believe it was FIU or FIU. There was a paper that we publish a lot of papers as collaboration. I think there was a paper that came out in December, maybe with FAU, I have to double check with one of our scientists. That's what our beautiful instruments look like. So this was my first this was at Amoco, kind of. This is what I started creating in the basement while I was still working and starting up the company to commercialize this product. This was our second big instrument. We received R&D 100 award for it. And then lots of other little things that came along. Just to expedite a little bit, it's already 4:30. I'll go through science just a little bit, just to show you what we do. This has been an incredible journey with lots of ups and downs. But very, very proud of what our team has accomplished. This particular instrument is the most published VCD instrument in the world. We do have competition now, but, and again, thanks go to people like Dr. Alva McConnell. This is our first installation in the pharmaceutical industry, which is Vice Pharmaceutical, became Pfizer. And that sort of jump-started us to other pharmaceutical companies. And it's like almost really like giving birth to a child. It was just incredible. There is nothing gives me pleasure more than to see scientists use our equipment. There is scientists from WISE, this is actually Oliver's boss, this is a director from BMS, talking about our technology at the ACS meeting. So there they mentioned how they use our technology, and it was published. Our users' work has appeared, and our work has appeared on covers of many journals, specifically several journals of medicinal chemistry, natural products. This is our work on fibrils and looking at neurodegenerative diseases. So we can see that the fibrils have either left-handed or right-handed form. Lots of fascinating work on ChemCom. Chemical and Engineering News ran a story on us when we first commercialized this instrument. And probably the biggest achievement is that we were able to move how scientists determine absolute configuration or change. Instead of doing X-ray crystallography, which is a gold standard, there's now probably about 8,000, if not more, molecules for which the shape, the absolute configuration, was determined with our technology. And so that's just an amazing feeling that somebody uses what you have brought to life. There are other applications. I won't bore you with that. If you're a chemist and you're interested by reading here, please let me know. I will definitely be happy to answer lots of questions. And there is, again, papers cover, so I can do that. The way we do this, just in case there are chemists in the audience, is we do the measurements and then we do the calculations and then we compare the two, and that gives us unambiguous determination of the shape. This is paper from AstraZeneca, a rapid alternative to X-ray crystallography. So that's really phenomenal. And lots of, I don't even have space to put all the pharmaceutical companies that now use our technology. And it's really making a difference. And I tell my family that we have never been busier than we have been from March. We have been really, I don't want to use the word bombarded, but lots and lots of molecules are coming in. I'm suspecting they could be antiviral. We don't know what they're used for. The customers don't tell us, but certainly VCD is helping. We are helping in the fight and in the finding better cure and better treatment. And I'll talk about vaccines in just a few seconds because that's a separate division for BioTools. VCD is accepted by regulatory agencies. We have 2 articles in US Pharmacopoeia, which is phenomenal. So specific articles, there are patents submitted with our technology for new molecular entities for pharmaceuticals. Lots of phenomenal academic use by very, very famous scientists and very famous institutions for all kinds of reasons. So we are very, very happy with what we have accomplished there. Our second instrument is a little less used. It's the reason, I guess, mostly from us because of how we sell these instruments. But we did get an R&D 100 award. It is used by pharma. It is in lots of phenomenal academic institutions. And what this instrument does is that provides the structure of biologics, specifically viruses, proteins, sugars, and nucleic acids. So you don't have to do, this was, electron microscopy or crystallography. You can also do chirality with that, but we sell it specifically for biologics. And the reason it's important is what I have been talking about is mostly targets, right? Small molecules, of which 80% are chiral. So we've just covered the targets. But if you look at proteins and diseases, a lot of proteins are also therapeutics. The ones you probably know best are monoclonal antibodies. It's a huge, huge market. probably 100 billion is not even the right number anymore. But it's very important. There is over 100 FDA. I kind of lost track over 100 FDA approved biologics on the market, including insulins and growth hormones and interferons and MABs and lots of them in development as well. And this is one of my favorite cartoons because in all of that development, in all of that formulation and development, formulation and quality control, one of the most important parameters for biologics is structure, because structure is very sensitive to perturbation of any kind and to physical state. And as you probably well know, structure is related to function. So we must know structure. And so here is two monks, and one of them says, forget enlightenment. I want you to concentrate on the structure of the protein molecule. So yes, structure is absolutely critical in all of the steps in folding, degradation, stability, and comparability studies. And we do that, again, with two of our instruments, the one I just showed and another, because we can get second and tertiary structure. Just like the first instrument, we have lots of customer publications. This was the first important paper from Amgen, from Dr. Lee's, a fantastic paper presentation from Merck, where they showed that our technology was a predictor of instability for monoclonal antibody earlier than all these other techniques, which is, of course, fantastic to hear because it's a very, very sensitive techniques. Some work from Bristol Myers Squibb. I know the time is coming up. I only have 2 minutes left, I think, before discussion. This is a product that was developed back at Amoco, so it's probably our most seasoned product installed in almost every company that used to do, well, I used to say that almost all insulins and all antibodies have gone through either our hands or this instrument. It's not true anymore, but it used to be. It's helped a lot of products get to the market. And the biggest applications are the ones I've mentioned: stability, formulation, mutation, and so on.
Excuse
so,
me.
Yep, I'll finish right there. This is my last
it's okay. I just meant to say that you can take another 5, maybe even 10 minutes. That's okay. You don't need to.
Oh, okay, because I like to leave time for discussion. This is actually my last two slides. We have a beautiful website where you can go. There's lots and lots of literature. I will say a couple of things about LinkedIn and social media because I'm speaking to lots of young people, I assume. My other recommendation to you for career, and I think you already know, all of you know this, is to use the social media appropriately and to really devote yourself to making sure that you have a really good profile and really good network on your LinkedIn site. If you don't have a LinkedIn account, I strongly recommend that. The best way to use it, one of the best ways is, for example, if you link into someone like myself who might have a bigger network than you and is busy, but maybe is able to help you, you can go through the connections and see what is it that you want to do and who is it that you want to get connected. And then you can ask for an introduction because it's much easier to do it through LinkedIn than to sit down and write yet another emails. I get over hundreds of emails a day. So for me, it's actually very difficult to do, or to call somebody and introduce you, or to even forward your resume to somebody, because I can't really think of who would be the best person. But if you know, for example, that you really want to work for Johnson & Johnson, or you want to work for Amgen, you can send me a direct message at LinkedIn and you can say, Rina, I noticed that in your network, you have blah, 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 blah as your connection. Would you mind making an introduction? And I can say, absolutely, I know this person really well. Or I can say, I have never heard of this person. They LinkedIn without my knowledge. Or like, I just don't know them, I've accepted. But that's a really good way to build your network. And building your network is very important. I cannot emphasize that, absolutely cannot emphasize that enough. So I think with that, I want to thank you. I do have lots and lots of other slides, probably 40 more slides, so I can answer questions. I wanted to leave more time, Lorena, so that I can have a discussion and answer questions on mentoring if there are any. I think a lot of my other slides are on women and women in science, so I can answer those questions as well. So thank you again for invitation, and thank you again for your attention.
No, thank you. That was fantastic. Very inspiring story for everybody. Also for me personally too, because I'm also not from the United States, come from another country. I'm actually from Brazil. And it was a nice flashback to my undergrad too in pharmacy school. So
oh, great. Fantastic.
Pressures there. Okay, so we have some questions. Let's get started with those. So one of the questions is, you mentioned that it's important to work on leadership skills. However, postdocs and PhD students are often very busy working on their projects in the lab. How can they carve out time to add more and yet, how can they carve out time to add more to their plate, like improving leadership and communication skills? So how can you, even though you have your project that you dedicate most of your time during your PhD or during your postdoc, how can they improve those leadership skills during that time? If you have any suggestions on that.
I think, Lorena, you can answer that question actually very well, because I can see that in you already. But of course, I'll be happy to mention my response to that. My advice to that would be spend less time on social media that doesn't matter. Spend less time, be more organized. Organizational skills, which I did not mention, are absolutely a key to your success. And it's not easy, but you can work on it. So the one thing you can do, LSD Hub is the other organization I was thinking of and was blanking out. During non-COVID time, that takes place once a month. So you can always find 2 or 3 hours once a month to be able to do something like that with the local society. The other thing you can do is maybe carve out an hour or two and go to local elementary school or middle school, if encourage women or even young men to enter science. So you can always carve out time. I know everybody is extremely busy, but if you really want to do it, my answer is that you will find time.
Okay. Yeah, just because you mentioned me and what I do and kind of turned the question to me, I guess, yeah, I agree with the organization thing and just setting aside a small part, let's say, of your time in a month. So for example, the Postdoc Association, we meet once every 2 months to discuss everything. And then we have these postdoc webinars every month. So, if you think about it, it's not that much of a time, but yes, if you don't organize and plan for it, it can take a lot more than that.
And the best way to do this, really, like if you join a committee, so for example, you're a neuroscientist, right? So if you join a committee for American Neuroscience Society, whatever the name of that society is, and you were on the committee meetings are probably once a month or once a quarter, you will carve that time automatically because that's on your schedule. So you will find the time for it. If you're just thinking about it, you will never find the time. Like I keep saying to myself, okay, I'll go get this done, but I never get it done because it's not on my calendar. Whereas talking to your group is on my calendar. So I will find time and I will do it. Yeah. And I'll organize my day to do it.
Great. Okay. Next one is, can you describe what type of questions you personally would ask a postdoc intern or someone that is applying for a job at your company? What kind of questions would you ask? So what we could expect if we're applying.
Wow, that is a great question. So to me, I think as a scientist, and maybe not necessarily always as a president of a company, so I cherish and I respect and like, or so to speak, 3 qualities. One is hard work, and I guess that comes because I've been working since I came to this country 40 years ago nonstop. Hard work, honesty, and dedication. I also like when people think outside the box. So to me, it doesn't matter if you're a neuroscientist and you learn those skills and you know nothing about spectroscopy, or you know nothing about proteins, or you know nothing about chirality. I can see after 20 years of business almost right away if a person is going to be willing to go an extra step to learn that. So my questions to you would be, my first questions would be probably, what did you like the most about graduate school? What inspired you to do this particular postdoc? What was the easiest and the hardest during your postdoctoral studies? Why did you not present, or why did you present blah, blah, blah? How many papers? So if you have a paper or two, was there an opportunity to write more papers, and what happened? Are you still writing? Do you still have data? So I would concentrate not necessarily on the skills that you got, but I will understand those skills from asking you, what is it that you did and how you liked it or didn't like it, and what you're willing to do. So are you willing to, for example, not work 9 to 5 and go to a customer side and be stuck at the airport? So that's also important to us. What are your communication skills with customers? So we, obviously customers is everything to a small business. And so your ability to communicate with customers is going to be critical. So if you're not looking at the eye during the interview and you're very, very shy, I might still hire you, probably will, for your scientific knowledge, but I will know not to put you for the customer communication. So, and if whoever asked that question, if you need more questions that I would ask, I would say, let's have an interview. Question.
Nice. What, aside from LinkedIn, how can we build the network during this virtual time? Do you know of any events or anything that's been happening for networking?
Yes, I do actually. So we did our first virtual conference last month, 2 months ago. I actually did 2 back-to-back. One was American Chemical Society and one was a smaller conference. And I would say in the smaller, and almost all of them give students and postdocs a very, very special price. And I know that it's not easy with the budgets, especially during COVID, but if you write to the organizers, they will let you in. I guarantee you that. Or you can write a company and ask them for support. You can do things like that. And I was struck in a smaller meeting, it was a bioprocessing, so it was all biologic site for BioTools, how you could actually go in and network with people, and they had networking sessions, and you get to know all these people. Even for us, you know, yes, we think about sales, but it's also for us. I was learning all about gene therapy because I was trying to figure out how can our technology work with gene therapy. So networking at virtual conferences is the best thing you can do right now because there's so many, they're inexpensive, and you can do them on demand, but you can still text, like you can use this texting capability of their conferencing to text all these people. And so that, I would say, is really a good way to do that.
Yeah. Okay. Adapting to the new
reality. Yep.
Yeah. Okay. So next one is, what skills were useful when you started your job in the industry, and which ones did you have to reinforce or improve?
Oh, excellent questions. I see you guys probably done this more than once. So definitely I was hired for the science because, as it turned out, you know, I went to interview, I wouldn't help somebody at Amico. I ended up being interviewed a few weeks later, and it turned out that my postdoc advisor was looking exactly for the science that I knew. So it just turned out perfect, which I never expected. So I was hired for the science, but I think I would have not succeeded would have I not been eager to succeed. Because you have to, as I said already, you have to put yourself out there. You know, people say, sit at the table, lean in, you know, sit at the table. But for for everybody, for men, women, you have to participate. You really have to. So the skill of being able to communicate with everybody and to participate is absolutely critical. You have to work hard and you have to make sure your job is done always, and that you report, and that you're compromising and that you are a team member. So when you go anywhere else other than your postdoc, and because you're now not by yourself, it's always a team sport, always. So ability to work with a team, and I come from the Soviet Union, so I do have like, sometimes like people say or maybe I've developed this like my way. I don't think it's my way or no way, but learning and really listening to other people is absolutely critical. I think we had to develop that. And on the side, and if you listen to my interview on LinkedIn, I always tell this story. So when I started the business, because I'm in physical chemistry, it's capital equipment, there was almost no women. I now know 2 others who started a similar business, but I still don't really know them. But every meeting starts with something, when you eat bagels and donuts, you will notice that men talk about sports. And so I had to adapt every country. And it's not just American, Japan, China, everywhere we went. We would go into the meeting and the first thing I hear are like, did you see that touchdown? Did you see that 4-shooter? Did you see that home run? And in the beginning I was like, what are they talking about? Like, what are they doing? So I adapted and I made a point of learning everything about the sport, whatever happened the night before, whatever meeting, whatever country I was in. So yeah, you adapt, but just be yourself and be eager to learn and be eager to help other people. It's not always about you. Be helpful and be true to yourself.
Okay. So since you mentioned the be willing to help other people and the teamwork and everything, so do you think there is less competition in the industry than in academia between people?
Oh, no, not at all. So I know for a fact, and this is still one thing that really saddens me, and I cannot change that single-handedly, and I know there is a lot of women working on it. If you're not a Ph.D., and especially if you're a woman, and I think it's true for men too, you almost cannot get to a higher level. You cannot get to managerial level, either scientifically or non-scientifically, unless of course you're in non-PhD required like HR. So if you're a scientist, you're not PhD, it's very difficult to get up. I'm sorry, what was the exact question? I was thinking about something else.
About competition.
Yes, competition. So for women scientists, it's hard, especially if you're not a PhD. If you're a PhD in industry, you still have to get up the ladder. You still have to publish. You still have, especially in industry, you still have to innovate. It's hard to innovate in industry if you're doing a team project, right? So you have, what I find is a lot of people try to find that new technique or find something else so that they can publish and they can go to meetings and they can get up above everybody else. So in industry, it's also very challenging because there are just so many people and everybody, from this level, there's only one. In academia, it's a different stress. You're by yourself and everything depends just on you. How many publications you have, how many students graduated, how many grants did you get? And you either made it yourself or you didn't. That's you and only you. In industry, you have all these team members who can push you up or push you down. So it's harder, I think.
Okay. Do you have any books or reading materials that you'd recommend to offer some inspiration or to show any particular skill development?
Oh my goodness, that's a great question as well. So I listen now to a lot of podcasts, either because it's harder for me to read and I look at paperwork the whole time. And I love a lot of aspiring podcasts. And as entrepreneur, my favorite is How I Build It, because it gives me an idea that I'm not the only one who didn't succeed in XYZ. But that's not for scientists. I think there's just a lot of aspirational leaders that you can find. I don't have a very specific recommendation. I try to listen and read as much as I possibly can. But I feel like, we succeeded very much with the science of the business. All these awards and recognition and all the people using, I'm not sure we succeeded in the business itself the way we should be 20 years later. Because I simply spend too much time on science, and I like to give everything away because I want people to use my science. So to me, listening to the business leaders, I watch Shark Tank, like I love all of that. But I also listen to NPR Science Podcast and just learning everything around me. There is one thing that I didn't mention, which I don't know if it's important for this particular webinar or not, but finding the right partner to me is a big deal. You can succeed on your own, and I never want to diminish a person succeeding on their own. But especially for women scientists, I think finding a partner that really will support and bring you up is critical. If you look, and I at some point I had this like a cheat sheet of all the very successful women, like Madeleine Albright and Indira Gandhi and Hillary Clinton, like the list, Marie Curie, and like the list goes on and on and on. What you will find is that there's always a very strong man behind them who did as much at home as the woman did, and who encouraged her to do her science, to do her work, to do her politics, and didn't take that away from her. And so to me, and I love listening to stories like that. So I don't know if that answers the question. I'm sorry I don't have a particular book.
Well, that's great. And then to finish, last question is, how do you slow down and find time to relax?
Oh, great question. I have two amazing kids who unfortunately have left home already. They're out, went to school here in Palm Beach Gardens. And so I used to watch dance a lot because my daughter was a dancer. So that was my biggest relaxation. And my son was a tennis player. So I used to do that. I turned to tennis now myself. So I love playing tennis. I love listening to music. I love going for walks. And during the virus, while coronavirus, I'm back to biking, cooking, organizing, redoing all of the accessories and BioTools, organizing offices, and spending time with my parents. So, but nice. Sports, music.
Yeah, that's very important, right? To find some time for yourself.
Great question.
Not everything can be business. So
yeah, exactly. It's important to relax and look at something else, movies. We have so much swimming. I mean, we live in Palm Beach County, we can, there's a lot to do. Swimming is probably one of my best relaxations as well.
Okay. All right. So that was our last question, and we are running out of time. So we're going to wrap up here. Thanks again so much for volunteering your time to speak to us and give us all this great information. Thank you so much for offering your contacts and helping us making contacts as well. Just offering your help in general. I think that's great, especially for us being in Florida, in Jupiter, or even anywhere in Florida nearby.
Not many places to get jobs here. I feel bad for postdocs here. There's not that many companies you can go to get a job if you want to stay right here in Palm Beach, or in South Florida.
Yeah, but yeah, it's great to have a point of contact, let's say. So that's awesome. Okay, so we're going to...
Thank you everybody very much. Thank you, Lorena, again. Stay that way, and all of you for joining. Thank you. You made your first step, and good luck to everybody. Stay safe and well.
Stay safe. Thank you so much. Bye, everyone.
Bye-bye.